So our next speaker is Dr. Tomislav Bacek. Tomislav received his bachelor and master's degree from the University of Zagreb, Croatia, and his PhD from Vrije Universiteit Brussels. And his research has been focused on the development of lower limb orthoses and their effects on assisted human walking. Since 2020, Tomislav is a research fellow at the University of Melbourne, where he's focusing on studying human biomechanics, human-robot interaction, and robot-assisted therapy, and has also recently become interested in computational modeling and its application to robot-assisted therapy. So please join me in welcoming Tomislav.
Thank you, Dana. What I will show you today are preliminary results, or some of the results of the preliminary human study that we've done at the University of Melbourne, which kind of what we are trying to. Yeah.
We can't see your screen. It's strange. We can see you. We can't see screen very well.
Yeah. Can you share your screen to Zoom?
Yeah.
I thought I was sharing my screen to Zoom. Wait, let me do. So do you see it now?
Yes, now we see it.
Yeah.
Thank you.
Okay. So what we are at the Melbourne Uni, what we are trying to understand is basically human walking, the mechanisms of human walking, and use this in robot-assisted gait therapy. Kind of what Gavin was saying. I think this comes nicely after his talk, because he's saying that all the rehab needs to be bespoke. You really need to approach each person individually, and so we are trying to understand on a deeper level what happens actually with each individual while they're walking. So this study is part of a project where we are trying to basically deliver or develop design framework to deliver optimal human-robot interaction based on model-guided and data-driven approach. What that means is that we do want to have some sort of model of human walking, but we don't necessarily need to understand everything through models. But then to complement this is data-driven approach, where we use data in real time that then using optimization techniques helps us to drive the gait into any optimum that we set. For the needs of this presentation, I'll separate or categorize human-robot interaction into two categories. You will have heard different phrases or names for these categories. What I will use is assistance-facilitated adaptation and assistance-induced learning. So the first one is essentially a case in which human benefits from the robot while is being assisted by a robot. As soon as you remove the robot, there is no benefit. So it's only useful while you are assisting the person. And this is what we have mostly today. You have robots that are either helping paraplegics to walk, or in industry, robots that are helping workers to do something, to do a particular task. As soon as you remove the robot, there is no benefit. The second one is assistance-induced learning. This is something that goes in the direction of restitution of human walking, where you're trying to use the robot to help the person while he's being assisted. But also the idea is that some of that benefit is retained when you remove the robot. That's basically motor learning. And so what we are trying to do with this study is basically understand human walking that can then help us to go in the direction of assistance-induced learning, because we really want to understand why humans walk in a certain way and how. So what we did, we designed a study where we had three factors: walking speed, constraints, and cadence. Walking speed, we have three levels. 0.8 and 0.4 are relevant speeds for the patient population. 1.1 is the normal speed. We started with 1.25. Marcus said 1.3 is where healthy adults usually walk. But because we have constraints, we saw, as you will see what constraints are, 1.25 was just too fast. It was unstable when we were walking with constraints. Constraints are just two levels. Either person is walking without constraints or with constraints. You will see shortly what they are. And cadence is just preferred cadence, and then two cadences that are higher and two that are lower. We use metronome to guide participants to walk at this any specific cadence. So that results in a total of 30 combinations, 3 by 2 by 5, and we ask person to walk 5 minutes at each. Now we group this. By frequency, step frequency. So we take any of the 6 walking speed and constraint combinations. So there are 6. And then we ask person to walk at all 5 frequencies for that combination without any breaks in between, which means that person walks 25 minutes without a break. And we have 6 of such bouts of 25 minutes. 3 are in one session and 3 are in the next session. So there are many combinations from these 30 combinations, but we wanted to avoid any potential bias. And biases can come if you have person walking with the orthosis 3 different sessions in a row, or if person is walking within the same session twice at the same speed. So to avoid all these different biases, after we've removed all these combinations that are susceptible to bias, we are left only with these 8 combinations. 2 for the constraints and 6 for speed. So if a person comes during first data collection session, we randomly choose one of the constraints combinations, which means that in the next session there is only one possible. And then for the speed, we also randomly choose one of 6. But then because of the way the constraints are essentially flipped, there are only 2 possibilities for the next session, which are these 2, but one of them has the same order of speeds. So to avoid the bias of the order of speed, we remove that one as well. And that gives us unique combination of constraints and speed in session 2, given the one that we chose randomly in session 1. In terms of sessions, first one is just baseline, where person comes in and gets familiar with walking, all the tests and the orthosis. So we always start with the free part where you don't wear any orthosis, walking at all 3 speeds that person will be walking at. And then we find preferred speed using the staircase method. And then person walks at that speed again for 7 minutes. This is what you see, the bottom row, 6 minutes, 2 minutes, 2 minutes, or 3 minutes, 2 minutes, 2 minutes is just a way of determining preferred frequency. You ask a person to walk for 6 minutes at their preferred cadence. Last 30 seconds, you note down that frequency. Then for in the next 2 minutes, first 30 seconds, you ask person to walk at higher frequency for 30 seconds and give them 1.5 minutes to converge back. You note down again that frequency, and then you do the same, but now on the other side. So the person walks at lower frequency, we guide them by metronome, and then they converge. And these 3 numbers are used to average and get preferred step frequency. After this part comes the orthosis part, so the constraints part, where the person is walking again at all 3 speeds, the same procedure, but person starts and ends the whole kind of part with the orthosis with 10-minute walking at their preferred speed to give them time to familiarize with the orthosis. So that's the first session. That's familiarization. And then there are 2 data collection sessions. They are both the same in terms of how they are run. We always start with 5-minute baseline metabolic cost, so we can measure your fatigue at the beginning. Then we have stair climbing and sit to stand activity, which is used for normalizing EMG. We don't use maximum voluntary contraction. We then also have walking at preferred step walking speed, which we measured in session 1. And this walking for 6 minutes is also, we have it at the end, so that we can compare the EMG metabolic cost at the beginning and the end of the entire session and see whether there are any fatigue or any changes. And then we have 3 25-minute walking bouts. So for 3 different speed and constraint combinations, person goes through all 5 step frequencies in a randomized order. Resting metabolic cost precedes each of these bouts because we use that metabolic cost to actually normalize the data. In terms of participants, I will show you the results from the first 2 participants. These 2 participants actually walk at 1.25 meters per second because that was the initial idea. But with them, we saw that that's too fast, and that's why we reduced to 1.1. And this is then what we've done with 12 people so far. In October, we will resume, and we hope to get another 15 or so, hopefully mostly female, because this part was mostly male. This is the orthosis. So basically what we do, we lock your knee, so you can't walk and person needs to compensate. This is just something we designed in the lab to accommodate for all the sensors that we are using. You can see Vicon markers on the person. You can also see EMG sensors, these black sensors are EMG sensors, 8 per leg. What you don't see is one on gluteus maximus. It's always the left leg that is locked. We can't lock it that the knee is locked at 0 degrees. There will be up to 7, 8, 10 degrees, depending on the person. Because of the soft tissue, we could not really stop that from happening. This is the experimental setup. I'll show you the video. So we have Monte Carlo lab at University of Melbourne. Person is secured with the harness so they don't fall down. They're also wearing a mask, which you will be able to see a bit. And again, orthosis and sensors. You can hear the metronome. So the person is guided by the metronome while we are recording EMG, ground reaction forces, motion analysis, so markers, and metabolic cost. Now I will show you what we've already seen, 3 visually very different compensatory mechanisms. The 2 participants on the left are the ones whose results I will show you. So this is preliminary study, and the participant on the right is part of the 12 people that is now undergoing the study. So on the left side, you can see what is known as vaulting. Look at the unconstrained heel. So the right leg heel, you see how the person is kind of lifting himself on the heel in order to lift the center of mass up so that they could progress their leg forward. So this is a very well-known compensatory mechanism in patients as well. The second one is essentially doing circumduction, which is basically walking very wide. You can see how the person is carrying the leg, left leg outwards in order to compensate. The person's got very wide step, and you will see later in the data, I don't know if you can see here, pelvis, left side of pelvis is also lifted a bit in order to compensate. So circumduction and vaulting are very typical of patients, and we were expecting this. What we were not expecting is the third compensatory gait, which is very common in patients, and this is a step 2 gait. This was one of the strongest participants that we had, and yet the person said that this was the most comfortable gait. It came as a very big surprise to us. We thought that this person would just be able to carry their leg forward, but the person said that they were trying different compensatory strategies, but this was the most comfortable. Now, in terms of data, I'll guide you through this. So this is for participant 1 that was on the left side. You can see in green is walking without the orthosis, in red is walking with the orthosis. 3 columns separated by white space at 3 different speeds. On the left, 0.4, in the middle, 0.8, and on the right, 1.25. Top row is step length symmetry, middle row is step time symmetry, and the bottom one is metabolic cost. So this is one person. If you look at the top one, step length symmetry, you can see it's changing a lot. The line that you can see, horizontal line at 51 or something, this is step length symmetry at preferred speed for that participant. What you can see is that across different speed, not only is the person not walking at that preferred step symmetry, but is also changing the direction. When the bars are higher than this line, the person is walking with a longer left step, which is constrained leg, which when it's lower, he's walking with a longer right step. You can also see variability is decreasing as the speed is increasing, which is also something we were expecting. If you look at the second row, step time symmetry, it's much more consistent than step length symmetry, which is something that the literature is divided on. Some people are showing that step time symmetry is what people keep constant no matter what. Some research has shown that that's not really the case. Our research has shown that does seem to be the case with both participants. At the bottom, you can see metabolic cost. As Marcus already pointed out, the faster you walk, the higher it is. There is no that U shape that sometimes it's seen in literature. You can see it in some case, but I think it's a bit a stretch to actually find a U-shaped curve describing metabolic cost. Second participants, pretty much the same. You can see very similar. Step time symmetry is much more consistent than step length symmetry. There is much bigger variability at lower speeds than at higher speeds. And the person is again changing direction of preferred step length symmetry, sometimes walking faster with the left and sometimes with the right step. With this person, especially at 1.25, you can see a U shape in metabolic cost, where the preferred or the lowest metabolic cost is at the preferred speed. Now, going back to the video that I showed you, you will now see the compensatory mechanism as quantified by these graphs. Just look at the BFJ, so the second, the row on the right. There is a lot of data here, but pay attention to dotted dashed lines and full lines. This is the difference between constrained and unconstrained walking. You can see that there is a noticeable difference in all these graphs. Now, this is the vaulting part. The ankle in blue, this is the difference in the ankle, right? The difference that you see here is circled in green is the difference in the right ankle, where vaulting is clearly increasing the angle, the plantar flexion angle of the ankle. In yellow circle, this is F figure. You can see the difference in pelvic motion. So the pelvis is actually being lifted more with constrained walking, which is something you would expect. And hip hiking, if you look at the J figure, is significantly increased. In unconstrained case, so the free case, the hip hiking is negative, meaning that the person's hip during walking is actually lower than during standing. While in the constrained case, it's quite the opposite. So it's a noticeable difference in hip hiking. In the case of the second person, so this person is not vaulting, but this difference here in the knee angle is how the person is compensated. So this is one of the ways, and the person is basically locking their knee more, which basically lifts their center of mass up. Similarly, pelvis is significantly tilted, significantly higher. And in terms of hip hiking, this person has also increased hip hiking, but in the free walking, their hip hiking is already, so the hip position is already higher than in the standing, which was the opposite for the previous person. And yet this person also further increases their hip hiking. Now, this is something a bit different, but something that we've also noticed. So if you look at the left, on the left side, knee angle, so this is 6 minutes of the same person walking under the same conditions. Literally nothing is changing, except the way person chooses to walk. So in red, and you can see this also in change in the joint trajectory, there is actually a knee flexion. So person is actually doing the weight bearing, the weight acceptance. In the blue part, at the bottom, you can see in the x-axis, the blue part is where the knee is essentially locked. So person basically locks their knee during weight acceptance. In the bottom row, you can see a different person, the same test, the same 6 minutes, the same condition, and the person keeps switching between locked knee and non-locked knee, right? Without us telling them anything. It's just the way they prefer to walk. So if you now look at the figure A on the right, and just compare the red and blue trajectories, this is knee angle during flexion, you can see exactly what I mentioned. The red one, there is flexion. The blue one, there is no flexion. Now, if you go down and look at the figure C, there is a clear difference in the red and blue trajectory of the vastus muscle. So the knee extensor, when the person locks their knee, is not active, basically. This is the blue line. When the person actually unlocks their knee and walks with the flexion, then muscle activity does appear again. Now, if you look at the B figure, this is ground reaction force in anterior-posterior direction. This is the forward direction. It doesn't change basically at all. Person is changing their gait, their knee flexion patterns, but the ground reaction force doesn't change at all. And if you look at D, temporal parameters on the constrained leg, which is stance time and step time, they basically do not change, right? So the functional descriptors of gait, the symmetries and anterior posterior ground reaction force, if you look at the maximum, do not change, and yet person is significantly changing their gait pattern, which goes back to what Marcus and Gavin already mentioned. Gait is really unique and personalized, and we need to approach it from each person's point of view. And just as the last result, so we had 12 people. So this is no longer preliminary 2 persons. This is from the 12 people that are actually now in the study. Unfortunately, we had problems with metabolic cost analyzer with 6 persons. So we didn't collect all the tests. So here we only showed 6 people that have gone through all the measurements. So we have metabolic cost for 6 people for all the tests. Again, green is without orthosis, red is with orthosis. You can see the speeds on the right is 0.4, middle is 0.8, and at high speed is 1.1. There is no metabolic cost, does not seem to be driving the gait. At 0.4, there is barely any difference, any noticeable difference. At 0.8, you can start seeing the difference. You can even see in the case with the orthosis that preferred step frequency seems to be the most energy efficient. While at the higher speed, which is 1.1, this is the most prominent. You can see the U shape, but you can also see large variability. So it's not clear, at least at lower speed, metabolic cost does not seem to drive the gait. At higher speed, it does seem to have some sort of effect. In terms of take-home message, I went through this with all the graphs in terms of step time symmetry, step length symmetry. So I wouldn't repeat myself. What I would say is, and we've seen this especially with that EMG graph that I was showing you, but also the way people compensate, it seems that it's just effortless for us to tap into different walking mechanisms when we walk, which is just providing further evidence. And we have no doubt that the remaining of the study will show pretty much the same, that we can't just focus on functional gait outcomes like gait speed and step symmetries, time, length, and so on, but we really need to dig deeper into muscle level, neural level, and really understand what is going on, how does each person walk, and why do they walk, because functional gait parameters are just showing us how they walk, but not why they walk. Just want to acknowledge everyone who's been working on this project. It's a collaboration between University of Melbourne, Imperial College London, and Monash University, and it's funded by the Australian Research Council. And with this, I will finish, and I'm happy to answer your questions. Thank you. So are there any questions? Yes.
No,
a couple. Can you just, so
yeah.
I can.
Thank you. That was very interesting and exciting, and I think it's the right. I mean, you're pointing towards the right question to find the right answer. My first question will be like, how would you choose the times that you have, like, when you design the experiment, for how long should each session be to avoid habituation? Like, how do you, based on what did you?
So how do you choose the 5 minutes?
Yeah, how do you choose that?
So obviously it's a very long study, and people are walking, they come for 3 hours during second and third session. So it was a balance between, we want to get as many different conditions as possible and still have enough data to actually measure the steady state. Now, there are studies, particularly with metabolic costs, which seems to be the longest to adapt, that show that 3.5 to 4 minutes people should adapt. That is, though, without any orthosis, right? So it's healthy. So have they adapted? The results suggest that they have, but we need to look deeper. For the kinematics and EMG, they adapt much quicker. The research is showing that for kinematics, sometimes it takes as little as 1 minute, and person is already walking steady state.
Okay. And the second one would be like, if you take a look at not the parameters, the scriptures, but like at the joint space, in all the data that we collected, do you have the feeling that, is there something that points towards this kind of individualization changes? Is there something that you notice in your data, or like, are there certain parameters, maybe body, maybe the the center of mass, I don't know, something that points towards this kind of individual.
Yeah, so we are measuring, you can see we're measuring a lot and we are calculating a lot. And so for some in the actual study, we also measure upper body. So we can also see the stability and so on. We haven't unfortunately done much analysis just yet. This is pretty much what we have done. But what you can see, and I think the graphs on it was this for both of them. If you look at just step length symmetry, right, there is so much changing. So the one person has no preference. That preference changes as the conditions change, right? And this basically is and it's the same for the second person, but it's obviously different numbers. But this is kind of telling us, well, obviously it's not even about it's about speed, it's about condition, and even for a single person, it's about in which condition that person is that will affect what is their preferred. So really what Gavin was saying, there is a progression of what person wants and needs at any given stage. And that's true even for healthy. Mind you, healthy have access to all the joints, healthy joints, they can do whatever they want, right? And yet they do choose strategies which are very similar to patients. That we find super interesting, but we need to look into. Okay, we have time for one more. Who was, I don't know who was the. Okay.
Thank you for your presentation. I have a question about how did you think about the foot drop condition? I mean, stroke patients, one of the main problems is not only that they are limited on the front movements, but also they are foot drop. It's quite a critical condition. But also, I think increase the metabolic cost. Do you think to extend your work to putting on this condition?
So the question is if we thought about adding, extending this to consider foot drop. So we were thinking of constraining ankle first, because we thought that constraining ankle would cause participants to compensate. Then we settled on the knee, because what we wanted to see was really if we can replicate. So the first idea was, can we somehow replicate the most common presentations in stroke, which is circumduction and hip hiking? And we did, right? Foot drop, we don't have any plans, because foot drop it's a bit different. Like how would you, what we can do, we could force the foot drop, right? All the time keep the foot down, so the person would need to compensate even more. We didn't think about it. What we did notice is that, I think I can say pretty much everyone, their muscle activity on tibialis, so the dorsiflector, is always high. So all of them, because of how they need to compensate, all of them are really trying hard to keep their foot as high as they can, because it's really like dangerous otherwise.
That's what doesn't happen with patients. Like they don't have a tibia muscle.
Yeah, I know.
And so it's like I think it changed a little bit. I think,
For sure.
When you drop foot or like you need more effort.
For sure. This is also, and this is like in that sense a limitation, but we are aware of, you can't directly translate this to patients. Patients have neurologically caused impairment, but like mechanical representation. We have mechanical representation and mechanically caused impairment. It's not the same, but it's very insightful. You can tell us a lot about, because patients didn't lose the same walking mechanisms that we have. They just don't have access to them. Okay. Well, I think, I think in the interest of time, we need to move on. Thank you for the questions. And yeah, we can move then to the next